Hello everyone, welcome back to MMBS111, Introductory Microbiology. This is mini lecture 8.2, and in this mini lecture we are going to be discussing transcription and translation, and these are the steps that cells follow in order to make proteins. There are several learning outcomes for this mini lecture. First, be able to identify structural and functional differences of DNA and RNA, define transcription and translation, name the RNA types and their functions involved in transcription and translation, explain the process of transcription, define codon, anticodon redundancy, and wobble, describe the process of translation, and explain how eukaryotic transcription and translation differ from prokaryotic. So transcription is what happens first in this process, and transcription is where the master code or set of instructions that's contained within the DNA is used to make an RNA molecule, and this is generally messenger RNA. And then that messenger RNA is translated by the ribosomes and used to produce proteins. There are some exceptions to this pattern. RNA viruses convert RNA to other RNA molecules. Retroviruses work backwards and convert their RNA to DNA. And there are a number of RNAs that are used to regulate gene function that are not involved in this process of making proteins. So there are three steps in the process of transcription. First, we have initiation, and this is where RNA polymerase is going to recognize a very specific sequence within the DNA molecule called a promoter. And the promoter is sort of a signal sequence that lets the polymerase know that, hey, there's a gene here that needs to be transcribed. Once that promoter has been recognized and that RNA polymerase has attached to the DNA helix, then elongation happens, and this is where the polymerase enzyme is going to work its way down that DNA molecule, and messenger RNA or some other RNA is going to be formed as the polymerase works down the sequence. The final step in transcription is termination, and this is when the RNA polymerase reaches the terminator in that sequence, and that DNA, that section of DNA is going to signal to the RNA polymerase, hey, that's the end of the gene, and then the polymerase will release from the DNA double helix. There are a number of RNA molecules that are produced in cells, and they each have different jobs. However, their structure is pretty much the same. So RNA and DNA are similar in structure to one another, except for a few differeces. First, RNA is a single-stranded molecule, not a double-stranded molecule. However, it is helical in its structure, so it does sort of twist up on itself. It contains uracil instead of thymine, and this doesn't change the code as DNA is transcribed into RNA. It still follows the pairing rules. So uracil, even though it is not thymine, still bonds to adenine. The sugar in RNA is ribose instead of deoxyribose. So those are the key differences between RNA and DNA. There are a number of different RNAs, as I said, that are produced in the cell. Some RNAs are regulatory structures and they help to regulate the expression of certain genes. Some of them are primers. So we talked about in mini lecture 8.1 how during DNA replication, we need an RNA primer in order to start the replication process on the lagging strand. So some RNAs are produced to be primers. Some RNAs are known as ribozymes, and these RNAs remove unneeded sequences from other eukaryotic RNA molecules. And then we've got these guys, and these are the ones that are really key for protein synthesis. So we have first messenger RNA, or mRNA. And messenger RNA is the molecule that becomes the transcript of the gene or series of genes from the DNA molecule. And it is read as a series of triplet nucleotides that we call codons. So 3 nucleotides in a sequence is a codon. We also have ribosomal RNA, or rRNA, and this is the molecule along with some proteins that forms ribosomes. And then finally we have transfer RNAs, or tRNAs, and these are the molecules that contain the anticodon. The anticodon is the match or the complementary bases of the codon for the messenger RNA. And transfer RNA is going to carry the corresponding amino acid for the codon in the messenger RNA sequence so that we can build a polypeptide chain or a protein. So once we have gone through the 3 steps of transcription and we have created this messenger RNA molecule, the next step is translation. And translation occurs on ribosomes. And we've already learned in earlier chapters that the ribosomes of prokaryotes and the ribosomes of eukaryotes are not the same. They differ in size. So the ribosomes found in bacteria, mitochondria, and chloroplasts are 70S ribosomes, and they're composed of a 50S large subunit and a 30S small subunit, while eukaryotic ribosomes are 80S, and they're composed of a 60S large subunit and a 40S small subunit. The small subunit is the part of the ribosome that is actually going to bind to the messenger RNA, the 5' end. This large subunit is going to join the small subunit once the messenger RNA is bound, and it is going to provide the enzymes and the binding sites for forming the protein and joining amino acids to form that peptide chain. So I mentioned before that a codon is a group of 3 nucleotides, and this sequence of 3 nucleotides, or the codon, is what determines the amino acid that is added to this growing chain of peptides. There are 64 different codons that code for 20 amino acids. So some of these amino acids are represented by more than one codon. And this is known as redundancy. And this allows for the insertion of the correct amino acid even when there are mistakes in the DNA sequence. So this is one of the measures that our cells have kind of built into them to help address things like mistakes and mutations. A wobble is, or what a wobble is, I guess, because amino acids contain, I suppose, a wobble. The first 2 nucleotides are required. These first 2 are essential for matching the codon and the anticodon with the amino acid. The third nucleotide doesn't change the amino acid. So this also allows for some variation or mistakes without changing the message. So you're still going to get the correct amino acid, even if the third nucleotide is not an exact match. So the way that the DNA code is interpreted to produce proteins is first we have these coding sequences within the DNA. DNA and RNA are read in these triplet sequences. And we start with the DNA and we produce a messenger RNA from the template strand. Okay. Then the transfer RNA anticodons are going to be the complementary codon to the messenger RNA codon. And that transfer RNA anticodon is going to bring with it an amino acid that is a match for the mRNA codon. So you see this first codon in the messenger RNA is AUG. AUG is the codon match of the amino acid F-methionine. The anticodon is the complementary codon or the complementary sequence. So A bonds to U in RNA, U bonds to A in RNA, and G bonds to C. We can use this relationship to help us make some predictions about what sequences should arise from, say, a DNA template, or what amino acid sequence is going to look like from a messenger RNA. If we know one code, then we can infer or work out the other codes. So transcription in bacteria occurs in the cytoplasm. It can happen immediately in bacteria because there is no nucleus. So in eukaryotic cells, transcription happens in the nucleus. And then the messenger RNA travels through those nuclear pores to get into the cytoplasm. Well, that's not how it works in bacteria because we don't have a nucleus. So once transcription has happened, that messenger RNA molecule is already in the cytoplasm, so translation can occur immediately. It also can happen with multiple ribosomes. So we call this a polyribosomal complex. It helps to speed up the rate of translation and increase how quickly proteins are made. So bacteria are able to produce proteins at a much faster rate than eukaryotic cells because their ribosomes can be working on the same messenger RNA simultaneously. This is the sort of general process for translation. So step one occurs when the ribosomal subunits join on the messenger RNA molecule and they create sites in the large subunit for transfer RNAs to place, to be held in place during this process. So once the ribosomes are attached, then we're going to have transfer RNA molecules that are going to be delivering amino acids. And the first transfer RNA molecule is going to enter this process at the A site. This is the aminoacyl site or the amino acid attachment site, okay? Then the transfer RNA, once that codon and anticodon have recognized each other, is going to shift to the P site or the peptide site. The next transfer RNA is going to take the old one's place in the A site. And that anticodon and codon are going to recognize each other. And once that has happened, a peptide bond is going to form between those two amino acids from the first transfer RNA and the second transfer RNA. And then translocation, or that movement, is going to happen again, and that second transfer RNA is going to shift to the P site. And this first transfer RNA is going to shift to the E site or the exit site. And that is how it will leave the ribosome. And a new transfer RNA molecule is going to find its place in the A site. And that codon and anticodon again are going to recognize each other. And that process continues until we reach the stop sequence. And then all of the components separate, the polypeptide is released, and it's going to go to the ER or go to the Golgi if we're talking about a eukaryotic cell to be modified, or it's going to be modified in the cytoplasm if we're talking about a bacterial cell, and then it's going to find its way to where it's needed. Okay, so transcription and translation take place in both types of cells, eukaryotic and prokaryotic cells. But there are a number of differences between the processes in these cells. So there's always a start codon. The start codon is always AUG, and this signals to ribosomes, hey, this is a messenger RNA, it needs to be translated. And where that translation needs to start. AUG codes for methionine in both cells, but the methionine in eukaryotes and the methionine in bacteria is not the same. So in bacteria, it's F-methionine. In eukaryotes, we just call it methionine, but they're not the same amino acid. Messenger RNA in these two in bacteria can code for several genes in a series. So we might have a number of related genes from the bacterial chromosome that all get translated at one time. In eukaryotes, messenger RNA only codes for one protein at a time. For bacteria, transcription and translation can occur simultaneously in the cytoplasm. So as transcription is taking place on that DNA molecule, as soon as there's available space for ribosomes to attach, it can begin the process of translation. So those activities can happen at the same time. In eukaryotic cells, that's not the case because there are not ribosomes in the nucleus, and since transcription happens in the nucleus, the messenger RNA actually has to be transported or moved from the nucleus into the cytoplasm in order for translation to happen. And then genes are also a bit different in eukaryotes and prokaryotes or bacteria. In bacteria, genes exist as an uninterrupted set of triplets coding for a protein. So there's no inserted unnecessary material. However, eukaryotes contain things like introns and exons. Introns don't code for anything. It's inserted genetic material that does not have a function that we know of. Exons are the sections of DNA that have the instructions that code for specific proteins. So before any of this can happen in a eukaryotic cell, we actually have to go through the process of transcription, and then we have to cut out the introns that don't belong and then put the exons back together. So there's some extra steps in these processes when we're talking about eukaryotic cells. And that is the end of mini lecture 8.2. So here's Some checking in questions. Make sure you go through these, double check that you're familiar with how you would respond to these questions. Also go back and look at the learning objectives. Make sure that you understand those as well. And here's our critical thinking question. So here's an example of something that you might see on an exam. If you are given this sequence as the DNA template, what would the messenger RNA sequence look like from this template? And then using the newly formed messenger RNA, identify the transfer RNA anticodon sequence and the amino acid sequence. So you're going to use this little DNA fragment to create both messenger RNA, the transfer RNA anticodon, and the amino acid sequence. And you can find the amino acid codons in this mini lecture. If you go back a few slides and find the amino acid codon wheel, that will help you to determine those sequences. All right, so hopefully this mini lecture was helpful.